This is in two parts. The first part is very short. It's about precision medicine. And I'm using precision medicine, really, a piece of evidence that we should want to have big pharma data. We should want to have lots of data, and we're going to need more and more data as time goes by. But then in part two, which is much longer, I will try to show that the situation is in fact much worse. And then I'll try and give you some ideas of solutions. So you're probably much more familiar with this kind of diagram than I am. The blue person here is the person that any given drug will help. So Abilify will help 1 in every 5 people. Nexium will help 1 in every 24 people. So it's prescribed to 24 people, but only 1 person is helped. And this is a product of the traditional way of understanding how medicines should be discovered and prescribed. This is the standard medicine effect. You have in the future, when we have precision medicine, we will be able to give drugs to those members of the population that the drug will help, and then we will have something more like 100% effectiveness. This is the idea. So the tailored medicine effect will divide populations, and we will require, therefore, a much larger number of subjects when we're doing clinical trials. And I hope you can see why that is. For any given drug, there are going to be a much smaller number of patients which that drug will help. We're going to need to find those patients, and for that we will need much larger populations of subjects to start with. We're going to need to do many, many more clinical trials with more and more precise targets, and each trial is going to be just as expensive as trials are now, perhaps more expensive, because you need to find more subjects, and finding subjects is a difficult hurdle, but also for other reasons. We're going to need to have much more data about the patients so that we can assign the patients to specific populations in order to carry out clinical trials which will test drugs which have a good chance of working for the members of those populations. All right, so we get down to this. 1 drug per person for any given ailment, or something like that. Maybe identical twins will be able to use the same drug. Now that causes a problem, and the problem, I think, can be expressed in a way like this. So it's not merely that pharmaceutical science will require ever larger quantities of data. Pharmaceutical science will require ever larger quantities of data squared because of this. And why is that? Well, because we communicate this will require ever larger quantities of data. Pharmaceutical science will require ever larger quantities of data squared because of this. And why is that? Well, because we communicate the results of scientific experiments by writing papers. And we're going to need to write ever more papers. That's perhaps just a consequence of needing to do many more clinical trials. We're going to have many more papers, but that's a kind of one-to-one correspondence. But where we get the squared effect is that most papers are false, and that's what I'm going to talk about. So there is a replication problem in scientific research. This is a gigantic scandal, which is going to require many more papers to be published. Maybe. We'll see that even that is not quite clear. That doesn't mean things are better than that. It means that there may be 5 already, which produced a series of arguments why published research findings are less likely to be true than we think. And this is true especially when we have studies in small fields, or studies where the effect sizes are small, or where there is a greater financial or other kind of incentive for the result to be true. So people like to publish papers which prove that what they believe is true. Or they like to publish papers which will make them more famous or richer or more hireable. And these kinds of interests are not necessarily concordant with publishing papers which are true. And Ioannidis showed this very clearly. A particularly bad problem arises where you're doing experiments and there are many alternative ways of analyzing the data arising from those experiments. Because if you do 1,000 experiments and collect 1,000,000 data points and you have 10 different ways of shuffling the data to support 10 different theses, and 9 of them come out in such a way that the data does not support them, but one of them comes out in such a way that the data does support them, you've just reduced the statistical likelihood of your conclusion being true by 1/10, because you've thrown away 9/10 of the relevant data, roughly speaking. I'm not a statistician, so don't blame me for anything which I say about statistics being false. All right, so journals too are incentivized to publish interesting findings which will make it about the journal, findings which will make people like the journal. All of this implies that published research findings are less likely to be true. The unpublished ones may be okay. Now, why are interesting findings likely to be false? Because interesting findings are findings which contradict what we all already know. And it's very hard to be interesting, but to be saying something which is contradicting what everybody already knows. I hope that is clear, why that is going to be hard to do. So if it's a battle between interesting and true, interesting will win. And does win. We know now that after the Ioannidis paper was published, we have many experiments trying to replicate scientific results, and the results of these replication investigations, we have many experiments trying to replicate scientific results, and the results of these replication investigations are really scandalous. Now, these scandals were first exposed in the area of social psychology and similar areas. These are, roughly speaking, areas where people have beliefs that they really want to be true. And I'll give you one example. It's a quite famous example. This is Amy Cuddy, who is a social psychologist at Harvard, very famous, very prestigious. She and a number of people she was working with put forward the idea that there is, as she says, a free no-tech life hack called power posing, which will help you get a job or convince people in an interview or overcome some hurdle on a first date, for instance. And the idea is that you increase your adrenaline, I don't know. So this is a...
So I'm going to start by offering you a free no-tech life hack. And all it requires of you is this, that you change your posture for 2 minutes. This is what we did. We decided to bring people into the lab and run a little experiment. And these people adopted for 2 minutes either high power poses or low power poses. They come in, they spit into a vial. We, for 2 minutes, say, you need to do this or this. We then ask them, how powerful do you feel on a series of items? And then we give them an opportunity to gamble. And then we take another saliva sample. That's it. That's the whole experiment. Here's what we find on testosterone. From their baseline when they come in, high power people experience about a 20% increase. The baseline when they come in, high power people experience about a 20% increase. And low power people experience about a 10% decrease. So again, 2 minutes and you get these changes.
Okay, and so on. It's a TED talk. It goes on for 20 minutes. It's a very good talk. She ends up crying because all the, particularly women she's helped to become more successful. They go for an interview. And she was on TV. She was teaching Stephen Colbert how to do power posing. And her co-authors fairly soon realized that the data did not support this effect. And Amy Cuddy still believes in it. she's modified her claims to some degree, but she is still a defender of the idea that there is an effect here. I try to do replication experiments in other fields, some of which are more relevant to pharmacy than the social psychology, although social psychology is not completely remote from the kinds of things that you're interested in this building. And it's been estimated that huge amounts of money are being wasted on scientific research which is not replicatable. Now, how does this work? Well, I've mentioned already that there is something which is called p-hacking. So you collect a lot of data and then you shuffle it around until you find some kind of result which will have a p-value that makes it statistically significant. This is called p-hacking. Another version is called hacking, which is altering the hypothesis after you know the results of your experiments. And each time you do something like this, you simply reduce the statistical likelihood that your conclusions are correct. The problem is that you do lots of experiments, each of which is honest in its own way. No p-hacking, no hacking. But you do 100 experiments, and all the ones which fail to produce an interesting result, you just leave in your drawer, and you only publish the one that produces a significant result. And you see that it's not a good way of getting statistically significant results. In other words, it's not a good way of getting results which are true. So autism is an interesting field, and there is quite a literature now on the way in which autism, and there are many results in the autism field which people would like to be true. And so people do experiments and they discover these results and get famous. They're in the newspaper. But then a year later they have to withdraw their paper because it was just too good to be true. And indeed it was not true, as was discovered when people tried to replicate it. So these are the things. This is the autism epidemiology checklist. So this is how you check whether an autism paper is no good. And it never is any good. Usually, statistically speaking, it's not likely to be good. So is there a misleading headline overstating the findings? Of course there is. That's the way you become successful. Are there unsupported assertions that autism etiology involves experimental factors? I have this in slightly larger type here. Is there a small sample size? Of course. Are multiple factors tested for the association? Yes. Is only one showing the association? Of course. Are the statistics corrected for multiple tests? No, of course not. Is there a replication sample? What's a replication sample? And so on and so on. So this is a well-known phenomenon, not just in autism, but in a range of other fields. It's a and so on. So this is a well-known phenomenon, not just in autism, but in a range of other fields. It's a well-known phenomenon in social psychology, particularly because that's where it started. But people are still doing unreplicatable experiments in social psychology and in autism standardly. So they're still doing it. Everyone in the field knows that there is a replication crisis, but the institutions haven't yet changed. And the institutions means the journals and the hiring committees. People get hired if they publish in good journals. Good journals don't like to publish negative results. Good journals don't like to publish boring results. Good journals need to publish, and people need to publish to get jobs. So the cycle goes on. Now, this happens in chemistry too. So there is one journal called Organic Syntheses. So it should be relatively simple to replicate, and they do it. Within 6 months of the submission, they guarantee that they will replicate your experiment or not. Now, the author of the paper too should find it easy to replicate the experiment. Organic synthesis is not rocket science. But it turns out that 7.5% of the submissions, this is in 1 year, 6-year period, they failed to replicate in this journal. This is the paradigm journal for believers in replication. And it is so because it's easy to replicate in this field. Yet even in this field, 7.5% of the submissions do not replicate. And the editor says, that we should see this as being a frightening fact, because in most other areas of chemistry, things must be worse, maybe a lot worse. Now, in the pharmaceutical field, the big drug companies, they either have labs or they pay labs to try to replicate the studies which they want to use as a starting point for designing new drugs. And so that we don't know much about the discoveries, but we do know a little bit. So we know that Amgen took a 10-year period of landmark cancer studies, and they tried to replicate them, and 11% of them failed to replicate. And then Bayer found that only I said that wrong. Only 11% of the landmark studies which Amgen attempted to replicate. Only 11%. And for Bayer, it was only 25%. So replication is difficult. It's expensive. If you fail to replicate, that does not mean that the result is unreplicatable. It just means that you failed. Maybe you didn't do it right. Maybe you didn't do exactly what the original authors did. Maybe it's impossible to replicate in drug trials because every patient is different. There are all kinds of issues connected with replication. We're beginning to understand more and more what these issues are. People are beginning to become more savvy about statistics. In the pharma field, people try to replicate because the costs of experimenting with a new drug are so expensive. It's a good idea even to invest large amounts of money in replicating before you start doing experiments in your company. All right. So what are the solutions? How do people conceive science in this? How do people conceive science in the future? Now, the first solution is to stop publishing. It's an obvious solution. Then you won't be publishing false results. This is not a joke. So it's the published research findings that are the problem. And the part of the problem is that we need to publish more and more precision findings. And precision findings already are going to have a low likelihood of being interesting. So if you find a new way of understanding autism that affects all autistic people, that's exciting. But if you can show that you can help just a little bit autistic people who are non-smokers and who have a certain gene and who have a father who didn't have a certain other gene, that's beginning to be less exciting. No reason to have an incentive for exciting results, and people can do proper science. And another reason why journals create bad incentives is because if you do really clever experiments and you get what look like exciting results, and which may even be true, then you don't want other people to publish before you do. And so you want to publish quickly, and you want to keep your data hidden for a long time in order to prevent other people harvesting even more exciting results from your data. And so the existing scenario creates incentives to keep data siloed, keep data protected. Now the power of big data, and remember the squared problem at the beginning, the power of big data arises only when it's shared. And remember the squared problem at the beginning, the power of big data arises only when it's shared. If you have huge amounts of data, but it's divided into 4 million little pods, then you don't have big data. All right. So there are people who seriously recommend that we stop publishing papers. We stop publishing. And one of the leaders in this field is Atul Butte, who is a very successful clinical translational researcher in San Francisco. He has his own institute for computational medicine. And he is a brilliant hacker. And his idea is, it's a waste of time for me to write papers. I'm going to collect data. I'm going to outsource my experiments to India. They will send me lots of data. day by day, and I will use my brilliant hacking skills. And he doesn't talk like this. He is very brilliant. I will use my new discoveries. I'll redo drug tests which have been carried out by pharmaceutical companies over and over again. I test the drugs much more thoroughly and carefully. And if I'm slowed down and I have to write a paper and wait for the editors to review the paper and so on, this is just a waste of time. Brings in all these incentives. I'm not interested in interesting results. I'm interested in results which will help people find the right drug in the right situation. So he is part of a campaign to get rid of this hurdle called publishing. Now, he's not being very successful, and he is still publishing. But another side of this is, even if you do publish, you should make all your data free. You should just throw it into the internet, all your data, even if you don't trust it yourself. Even if you don't trust it yourself. And you should describe it in such a way that people can find it, advertise the existence of this data. Maybe people will be able to find jewels in your data that you couldn't see. And this is what I recommend. I'm assuming that some of you are involved in doing scientific experiments and will be involved in doing scientific experiments. All of my friends just take it to be a matter of course. It's obvious. You should publish all your data. You should deposit it in the repositories which the NIH is creating for all kinds of data. And you will get rewards by becoming known as somebody who publishes, deposits their data. I don't know how it is in the pharmacy school, but in the medical school, thanks to me, publishing your data, so sharing your data, is a criterion for tenure, to become tenured. So getting grant funding is one item, publishing is another item, sharing your data is another item. Another reason why you should share your data is because your data may be of value only if it's combined with certain other data that you don't have, but which other people do have, and which they can see the value of your data in terms of their data. And you can see that this could be a snowball effect. It is a snowball effect. All right. So the next solution is, these things I just want to say in general, these attempts to solve the replication crisis are gradually transforming science. So Atul Butte is one example of the way science is being transformed as a result of the replication crisis. He is a harbinger of the future. But there are four other ways. The first one is what are called reproducibility, harbinger of the future. But there are four other ways. Is what are called reproducibility projects. And the most famous of these is a cancer biology reproducibility project. There was also a psychology reproducibility project. This is the most famous. The idea was to take landmark studies in cancer from high-profile journals and then try to replicate all the experiments that were performed. And this may mean replicating at the bench in the lab, or it mean replicating the computational statistical treatment of the data coming from the lab. And the results of these attempts were, as they say, muddy. That is to say, a few studies could be replicated unproblematically. A few studies could not be replicated clearly, but in the middle you had a lot of uncertainty as to whether there was replication at all. And this is because the studies themselves were scientifically very complicated. The strategy was to have people from the original team part of the replication team so that they could have a better understanding of the methods used, and they could replicate the methods as closely as possible as that scenario would allow. But still, you never know whether you're replicating as closely as possible, because there are all kinds of hidden presuppositions that experimenters bring to the lab when they perform experiments. And we're going to talk about that very soon. So we mentioned organic syntheses. Now, a replication is normally much harder than it is in the area of organic synthesis, as I said. And there are two ways in which replication can fail, or two reasons. One reason is that it is innocent an innocent reason. So in clinical trials, it's hard to replicate because patient populations are different, they're always different. They're likely to be different in ways which affect the result. And that was an issue for the Cancer Reproducibility Project. But then there are non-innocent reasons, and that's what I'm primarily interested in. And this is where ontology comes into the picture. So the innocent reasons have to do with the desire, which may be conscious or it may be unconscious, to keep your data secret. Now, the NIH currently has a rule that you have, if you do research which is funded by the NIH, you have to publish all your data within 1 year. That means share it. That's what all the repositories are that the NIH has created. And there are similar rules in other countries with equivalent institutions. And the general rule now is that you have to publish data within 1 year if it's funded by any kind of open access way, so anybody can read it. They don't have to subscribe to the journal. And it also means publishing the data, which means, for instance, supplementary data which may go on the website of the journal publisher and which could be accessed through URLs which are provided as part of the data, so that people can check that you did the statistics right and so on. And the problem is that you can't check, typically. And the reason why you can't check is because everyone describes their data in slightly different ways, sometimes in very different ways. So when you create a database, you use column headers. And the reason why you can't check is because everyone describes their data in slightly different ways, sometimes in very different ways. So when you create a database, you use column headers, and typically the column headers will be codes. And you may know the meanings of your codes, although that is not always true. But other people don't know the meanings of your codes, and people will publish the data, but they won't always publish anything like a data dictionary, which would enable a third party viewing this data from the outside to work out what it's about. Now, brilliant hackers like Atul Butte, they can solve that problem. They just are brilliant hackers. But there are very few people like Atul Butte in this world. And the result is that data, even when it's published, even when it's shared, even when it's deposited in an open access repository, is not always useful. Now, ontology is a discipline, I don't know, I guess primarily a computer science discipline, although ontologists tend to be people who know something about computer science, but then something about something else, such as biology or medicine. I'm a philosopher, which means I know everything, but nothing else. So I don't know anything about pharmaceutical science, or I do know a little bit about medicine now, but that's because I've worked with people like Atul Butte on projects to try and make data sharing work better. So ontology is a discipline which tries to bring about a situation in which, even when people use different sets of codes in order to describe their data, outsiders can still understand what the data are about, because the codes have been labeled or tagged or annotated with words in what is called an ontology. Words or phrases. And the words or phrases, nouns, roughly speaking, at least a large number of people in the relevant discipline to describe the entities which the data are about. And so it's a little bit like a lexicon, but the difference is that these words in the ontology have logical definitions, which means that it's possible to use the result of tagging a database with ontology terms to reason about the database. And if you do it right, you can then put two databases together, and because you've tagged them both with terms from the same ontology, you can combine them into a single database. And you can keep adding more data, and then because you're tagging all the data with common ontologies, then the data will be able to be reasoned with across many, many databases in a way that you can't do now without brilliant hacking. Now this methodology arose in the wake of the Human Genome Project. The gene ontology is not an ontology of genes, it's an ontology of old biology. So it contains terms like cell differentiation or death or cell division or protein binding. And these terms were used to tag gene sequence data, or more generally to tag gene product data, so proteomics data. And now there are millions and millions of data items which have been tagged using gene ontology, which means that all the data in those millions of artifacts is accessible through this one resource, which proved to be an immensely useful tool, and which is used nowadays by tens of thousands of people and by every drug company, for instance. The drug companies gave money to the gene ontology at the beginning. All right, I'll come back to the gene ontology later. So the next solution is, I'll come back to the gene ontology later. So the next solution is to require pre-registration. There is something called the Center for Open Science, which had a pre-registration challenge. The idea is that before you start doing experiments, you have to pre-register your hypothesis and your method, and then your design and so forth. Then you start collecting data, then you start analyzing data, then you publish the data, and then you receive $1,000. And all you have to do is to pre-register your hypothesis before you start the experiment, and then you get the $1,000. And there's $1 million waiting there. Maybe some of it's gone already, but now the idea of the registry is that now you have an audit trail, so people can't put experiments into their drawer if they fail. They're going to keep track of all those hypotheses, and the ones that fail are going to be added to the statistical store. It's going to remove redundancy, because if everyone has to register their experiments, you can check whether given experiments have been performed already. And the idea, and this is another place where ontology steps in, the idea is that you will guarantee reproducibility, because you're going to force people to, when they register, to specify the hypothesis, methods, the statistical models. Everything which is needed to ensure reproducibility of an experiment is going to be registered in advance. Now that we don't have that at the moment, for reasons which I will make clear in a minute. So you have to specify all of these. And this is a bit like the FDA requiring all clinical trials to be pre-registered on clinicaltrials.gov. Now the problem is that the clinicaltrials.gov is clinicaltrials.gov. Now the problem is that the clinicaltrials.gov is a little bit different, but I think in this respect it's the same. The Center for Open Science still does not impose standards on how hypotheses and methods and results are to be described. So you still have this leakage problem that people describe their methods using their own secret codes, or using terms which are too vague, too general, too local to enable outsiders to do the replication. And the kind of example which I don't know whether this is even readable from out there, but these are 62 different labels used in different research communities for one protein. Now, many of you, I guess, know a lot about proteins, and you can look at all these labels and you can say, oh yes, that's just cytokine A15. But these have 61 other ways of referring to cytokine A15. And even cytokine A15 may be A with a big letter A or A with a little letter A. And of course, human beings can deal with that, but computers, they are really stupid. All right. Then another solution, which is propounded by Ioannidis himself and by his acolytes, is change the p-value threshold. So instead of 0.05, it will go down to 0.005. So then you need to work really hard. You must have to have bigger sample sizes. That means bigger data. So not merely more precision studies, but for each study, you need to have bigger data. You need to have millions of subjects. And that's happening now. More and more experiments are engaging study data derived from millions of subjects, through consortium-based research, where the same experiment is run in dozens of different institutions. All right, I said that. So 2.7 million samples were genotyped by this particular. This is just an example. And then finally, or this in summary, I guess, since this is not something new, sharing of data through global science, consortium science. And so this is one example of this, the Digital Transformation Initiative, which is designed to bring about a sharing of data in chemistry and materials science, and through something called pre-competitive collaboration. So this happened, I believe, first of all in the European Union, where the EU gave money to the pharmaceutical companies for would no longer be competitive. It would just be accepted by all of them and made freely available to all of them. And thereby you save money, but you also increase the size of data that each of them have available to do research in more proprietary areas. And then there are these other examples of pre-competitive collaboration. And then again, how do you make all of these gigantic bodies of data which are shared between different companies, how do you make these data discoverable if they're all described using their own different codes and so on? And it's at that point again that ontology comes into play. So ontology is taken somewhat more seriously in Europe than it is in the United States, because governments are more involved in healthcare for one reason. And so people are more willing to accept standards for describing things like data. Not always very good standards, but still they're willing to accept standards. And that means that they're using ontologies in describing clinical. To accept standards for describing things like data. Not always very good standards, but still they're willing to accept standards. And that means that they're using ontologies in describing clinical and other kinds of data. And the gene ontology I already mentioned. Now, I guess, how much longer do you want me to talk? I can go on like this for hours.
Just keep going. 10 more minutes.
Okay, 10 minutes. You tell me when I'm done. So the gene ontology I said was I'm going to hand out my handout now. So it turns out that Buffalo is one of the places in the world where this kind of ontology research is performed most energetically. So I'll price this out all the way up here. Can you take care of those? I don't know if I have enough. I'm saving a few behind in case people don't get one. So this is a list. It's not quite complete of the ontologies that were created or maintained, are being maintained in Buffalo. So the gene ontology was created in England in the European Bioinformatics Institute and in Stanford in 1998. It's the oldest ontology in the modern sense that was really successful. An ontology which is much more successful is in your iPhone, because the Siri system in your iPhone is based on ontologies, and the CEO and CIO of the company, the Siri company which created it, is also one of the leading ontology figures from the 1970s, 1980s in Stanford. But the gene ontology is now maintained in ontology, for instance, is here in Buffalo in the Department of Biomedical Informatics. His name is Deal. And I'm mentioning this for Alexander Deal. I'm mentioning this because if any of you are interested in getting involved in this ontology work, then there are a number of people. You can contact me or you can contact him. A number of people here in Buffalo who are doing ontology work which may be in areas which overlap with your areas of interest. So neurology is a particularly strong field. Immunology is another field. Psychiatry is another field. Dentistry is another field where we have ontology personnel here in Buffalo who are quite prominent in the world of ontology because we just do a lot of ontology here. All right, I'll come back to this list later on. So because the Go was there, you could gain access to the genomic data you needed very easily because the gene census lexicon. So it advanced discoverability. Now, if you can find data relevant to your needs, so data about development of tumors in pediatric shrimp. You can find it very easily using the gene ontology, filtering on species, tumor, and young. You can do that for any combination of terms. They have a really powerful filtered search, and they have a huge database and a huge body of literature which are tagged using this set of terms. But now you use it, and then you realize how good it is, but you also have a body of literature which are tagged using this set of terms. But now you use it, and then you realize how good it is, but you also realize that if you have your data tagged with gene ontology terms, then your data will become more valuable. And so the body of data which gets tagged with gene ontology is growing all the time because everyone wants their data to be discovered. Not because it's interesting. The interestingness doesn't come, no one is interested in tumors in pediatric shrimp, except 3 people. It's not interesting, but still, if you're working on it, you want your data to be discoverable by them too. All right, and it leads to the integration of the tag data. So this is the data, and this is the gene ontology. And the data about membrane-bound organelles is going to be somewhere in this hairball on the left, and you can find it very easily just by going to the gene ontology browser. The Amigo 2.0, you will find it very easily in Google. Now the next ontology I'm going to talk about very briefly is called CHEBI. CHEBI stands for Chemical Entities of Biological Interest. And as you can see from this, it's a reference database for chemistry. And we don't work on CHEBI in Buffalo. We work with CHEBI. We have friends who work on CHEBI. There are various other chemistry ontologies. There's a physicochemical process ontology. There's a chemical information ontology, which is about information artifacts about chemistry. There's a statistical methods ontology. There's a chemical methods I think I mentioned that. And this is where we were before with the pre-registration challenge. So the idea would be, and they could do this very easily, tag their hypotheses using the gene ontology and the other relevant ontologies, tag their methods using the chemical methods ontology or the biological methods ontology, which I'll refer to in a minute. And if they do that, then everyone at least will have some common basis for understanding what they're talking about, together with definitions, which are logically structured, but they're in English. You can understand the definitions. So this is the idea. This is how ontologies could help solve the problem of all of these efforts to solve the replication crisis by requiring pre-registration of hypotheses and methods. Now, so this is CHEBI again. I keep coming back to CHEBI because I want to get to this ontology, which is an ontology we developed here in Buffalo, and it was developed in conjunction with one of the people working on CHEBI in England, a lady called Jana Hastings down here. England, a lady called Jana Hastings down here. So she was working on CHEBI, we were working on emotions in connection with psychiatric research, amongst other things. And of course the two are intimately connected. And if you don't see why, then I'll go out for a drink with you later on and explain. So the emotion ontology consists of emotion terms like feeling in control, feeling nervous, feeling bad. These are all feeling terms. Physiological response terms, breathing at a slower rate, muscles becoming tense, and so on. And these are linked then to chemistry terms. So we link the chemistry ontology, the gene ontology, and the emotion ontology to tell you about the effects of dopamine, for instance. It's it has the role of neurotransmitter. This role is realized in neurotransmitter receptor activity, which is part of that thing we call happiness. Now, we've done this for many kinds of interesting chemical phenomena, drug addiction and so forth. This is just one example of the way you can use ontology. This is just one example of the way you can use ontologies to capture information which normally would belong to separate disciplines which are not linked together, to very easily capture the information and link it together in useful ways. All right, that's more on Cuddy. This is about how Cuddy is related to the gene ontology. We saw this earlier. Now, the reason why this is important is because we were part of developing a solution to this kind of problem by creating a registry. Now, there is a very large antibody registry, which is supported by NIH, and we work with the people, primarily this person, Anita Bandrowski. So the idea is that when people publish papers, they are often sloppy when they describe the chemical compounds that they use. And if they want to be sloppy, they should be required at least to link to an entry in a registry, which would give you a standard way of referring to a given cytokine, for instance. And that's what the antibody registry is there for. The antibody registry is huge. It's a gigantic database of antibodies. Each antibody then gives you, each entry in the database gives you a unique ID number and a preferred label, but then it tells you how to find your own particular way of referring to that antibody and how to label it in a standard way in order, and this is the idea, in order to satisfy NIH funding rules. So the idea would be that you only get your funding if you agree to use registry entries when describing antibodies, for instance. And now, we created the Antibody Ontology, and the Antibody Ontology. And the Antibody Ontology provides more than just a unique label. It provides also information about the targets associated with the antibody, information about conjugations, information about vendors, catalog numbers where you can order the antibody. Now this is important because antibodies the same antibody, chemically speaking, which you buy from one vendor in 2003 might be doing very different things as the same antibody that you buy from another vendor in 2017. And if you're going to keep track for replication purposes of what you did in a given experiment, then you need more than just a unique label. You need a unique label which takes you right to the batch, potentially, and which tells you also about what's been discovered about antibodies from this batch as contrasted with antibodies from another vendor and another batch and so on. So the antibody ontology it was created by Alex Deal. It's a Buffalo artifact, and it's also very large. It's smaller than the NIF antibody registry, but it's much richer in terms of what you can do with it. And it's primarily focused on antibodies used in immunological experiments. Now, I can talk a lot about statistics. So statistics packages don't always work as it's written on the tin. And you don't always know which statistics package people are using and which version and so on and so on. And think they work one week and then they don't work. And so I'm part of this problem, and it gives you a set of terms for describing statistical tools, statistical processes. Again, you can associate with those terms in your database how successful your use of it was, what you used it for, what you did with the data. In other words, what packages could ingest the data which were outputs of your package that we're using, and so on and so on. So I won't say any more. I have a number of other such ontologies which I could describe, but I think that's probably enough to be going. Okay. Thank you.
Thank you very much. We'll now proceed to open the floor for questions. I can't find the mic.
Is that the mic?
Oh yeah. It's quite big actually. So I guess we can be here all day long asking questions based on the talking, the variety of topics. You want to go first? All day long asking questions based on the talking, the variety of topics. You want to go first? Yeah. Oh, I forgot. Maybe students first. So you guys can have the privilege. Oh, all right. So you're gonna have to wait for me.
Yep.
presentation. I was just wondering, for the methods that used for adulterating published research papers, for harking, would that be considered a crime? Would it be lying or cheating because you're altering the original hypothesis, because that was not the original goal?
It is not lying or cheating because it's not required that you state your hypothesis before you do the research. But it should be required. And once it is required, then people who do harking will, once registration or something like registration is required, people who don't register will be able to be accused correctly of lying or cheating. So it's a question of raising the standards of scientific research in order to solve a problem with scientific research, which is affecting the reputation of science. And if science is going to continue to be supported generally by the wider public, then science should insulate itself from the kinds of scandals that we see. That's the idea.
Right, thank you.
Rob, you want to go ahead and answer?
Sure. So I was curious. So this is really fantastic. These frameworks, but what are the? Could you articulate maybe challenges in curating information that's comparable when you put them into these categories? So if you're trying to create if you're trying to connect a large series of data, especially clinical data.
Yes.
I guess how can you be assured that the information in the category that's combined is actually comparable? It could be, experiments done differently.
Yes, so one way in which this problem is solved is that there are so many people using some of these ontologies. Not all of them are used to the same degree, but some of them are used a lot, which means that there are lots of eyes on claims which are made about the compatibility of this annotation with that annotation. Ability of this annotation with that annotation. So people learn what annotations survive critique, and they use those annotations as time goes on. This is part of a larger virtuous cycle. At least we hope it's virtuous. So the gene ontology is used to tag data, and that's done still primarily manually. There are teams of PhD biologists whose job is to curate literature. And what that means is they read papers and they say this paper is about a gene ontology term, or cell division or apoptosis or something. So that paper has been tagged as being about apoptosis. And then there is data in that paper about apoptosis in some particular kind of context, in some particular kind of organism. And so they use context terms or organism terms to tag that data. And the tag X, but there is no term for X in the gene ontology. And they send a message to the gene ontology tracker. And I believe the guarantee is that there will be a response within 48 hours. And the message might say, I need a term for X. And the response might be, well, we call X Xing. And so you should use that. Instead of just saying X, if you call it X, then you'll confuse people because X is too unspecific. So that's, but sometimes they say, yes, you're right, we need the term X in the gene ontology and we've put it here. And it's changed every night. The gene ontology is updated every night. Now that's the editor's version. The release version is every 6 months, but the editor's version is constantly being updated. Yes?
So thank you very much for that talk. It's good to see you again now. So I had two questions. One was a follow-up on Rob's question here of actually curating, the need for curating an ontology, partly because I see that process as not being particularly scalable, given the dynamic nature of everything around us. And so I assume at some point much of the work could be done through natural language processing, or at least developing a narrow set of tags through completely automated methods.
So this is an interesting area. If you'd asked me this question 5 years ago, I would have said with confidence it must be done manually, because all the experiments that we've seen people performing trying to do tagging computationally produce results which are much less useful. And that was true, and I think I understand the reason for why it was true. Machine learning is based on statistics, and the primary use of gene ontology when genes are overexpressed or underexpressed in particular kinds of tumors or whatever it might be. And so if you use a statistical method to create a statistical baseline for another set of statistical research, then you'll end up with the baseline won't be a baseline. It will be too arbitrary. Now, machine learning has progressed quite a lot in the meantime, and there are one or two examples of successful use of machine learning to do tagging. And so I'm beginning to weaken in my skepticism. I'm not yet willing to throw the manual curation industry overboard, because there are hundreds of people now whose daily job is being curators, data curators. I'm interested, so I think that I'm also interested in the problems on the machine learning side, so whose daily job is being curators, data curators. I'm interested, so I think that I'm also interested in the problems on the machine learning side. So I think the problem which I find when I'm talking about ontologies in other areas, which is the other areas which are not just more nuanced than biology, but also changing even more rapidly, changing on a daily basis, because groups of people of one sort are moving to a new village or something. The problem is creating gold standards. So to do machine learning properly, you need to create a gold standard. And it is very expensive. And if you have 1,000 parameters that you need a gold standard for so that you can do machine learning in a reasonable way, you need a really big gold standard, and that has to be created manually, and then you're ending up with an unscalable problem of gold standard manufacturing. Now maybe you have a solution to that gold standard problem.
So I wanted to actually take up, play devil's advocate on one of the things you proposed about an a priori hypothesis as being a prerequisite for potentially the file draw problem or some bigger issue in replicability. It's possible that a hypothesis, a priori hypothesis strategy might be too simplistic given our ability to collect data. So for example, you might send out a weather satellite out there to just go observe and get data. And your goal might be to eventually model or develop a model of reality as a consequence, not necessarily to test your hypothesis. You were really never interested in the hypothesis.
Yes, so there is a lot going on before people formulate scientific hypotheses. So you may have a certain hypothesis about how hurricanes are formed, or earthquakes, or something like that. And you collect a lot of data, and you puzzle over the data, and you create a hypothesis. And there's nothing wrong with that. Provided you don't say that the evidence that led you to formulate the hypothesis proves that the hypothesis is true. What you then need to do is to say, my hypothesis is that earthquakes are caused by hurricanes 5 years prior doing such and such. And then you are going to register that hypothesis, and then you're going to look at the weather in the future. You can't really do experiments with hurricanes or with earthquakes, but the hypothesis-driven science. So there is observational science, where you are just observing phenotypes, for instance. But hypothesis-driven science, which is what the NIH and the NSF pay for, that's their mantra. We need a hypothesis, and then we need a method, and that has to be done on the basis of something like pre-registration. And pre-registration, I think, this is a big gap in the NIH's own new regulations on replication. Pre-registration has to involve the use of controlled vocabularies with definitions of the terms. And if you're going to do that, you might as well use ontologies because they give you the logical extra capabilities of testing consistency between data that's been annotated and so forth.
Chris Daly, I'm from the Pharmacy Practice Department. So
okay.
my question will come at a little bit different angle. So I found the comment you made about the EU paying to have the pharma companies submit their data to a central repository rather interesting. So our work is involved in our interventions, right? Trying to improve patient care and make that translatable over multiple settings because we're seeing pharmacists do different sets of activities. So one issue we're finding is trying to establish equal playing ground for more data sets. And we're limited at the university in what we can actually do because we can't practice that here at the university, so we depend on external partners in order to do that. So we're having issues getting external partners to A, access data, but also to mention what you said about the compete points. And if you're going to relinquish that statement on not competing on safety, and the reason for that is our institution, CMS, Centers for Medicaid and Medicare Services, have established that criteria that they're graded on based on the safety parameters. So there's an issue that we're trying to solve and get through. So what are your thoughts on that?
So first of all, the European Union was the first to introduce, I think, the first to introduce these kinds of pre-competitive data sharing agreements. But my understanding is that there are similar agreements also now in the US. But I imagine that they're focused primarily on the chemistry side. So this is data about molecules, basically. There are people who are lobbying. I think that I was lobbying today. So lobbying in the sense that I was lobbying by giving talks to scientific meetings, and people who are lobbying. I think that I was lobbying today. So lobbying in the sense that I was lobbying by giving talks to scientific meetings and the like for greater data sharing, also not on the chemistry side but on the patient side. And there is, I didn't mention it, but there is an ontology for adverse events, for instance, which is designed precisely by people who are trying to bring about a situation where data such as the data you described would indeed be not merely shareable but discoverable because it's tagged using the ontology for adverse events. And I can put you in touch with the people who built that. I'm one of them, but I know nothing. I can put you in touch with the people who built that, who are actually using it to do research on adverse events. Some of it in conjunction with MEDRA people. I take it you know what MEDRA is. Okay, so MEDRA is an organization which I believe works for the CMMS, is said, but it works with other organizations too, and it's an organization for recording adverse event data.
In clinical trials or post-market?
I don't know. But certainly if you don't know, you should know. Because that would be one place to look. So if we talk afterwards, I can give you the name of somebody who would be able to answer these questions much more intelligently than I can.
Any other questions, please?
That, a protein behaves differently from the others in that same family. You want to separate that out. Is it difficult to take data out that's already in the
Oh, that's a good question, yes. So that's a problem which, again, the gene ontology was the first to address, and they found the way to address it, but it's such a difficult problem that it's hard to address it thoroughly and correctly over a multi-year period. But it goes like this. You invest lots of effort in tagging all your data using an existing version of the protein ontology, which is another ontology maintained in part here in Buffalo. And then 2 years later, you need to make a change in the protein ontology like the one you just described. And then all that money you spent tagging with the old protein ontology may no longer be yielding any value because the protein ontology itself has changed. And the way we solve that is by giving each version of the protein ontology its own unique number. The ontology itself has changed. And the way we solve that is by giving each version of the protein ontology its own unique number. And then when you tag using the protein ontology in any given year, you know which version you used. And the changes are usually additive. In other words, you add more information, you add more branches to the ontology. So those changes don't reduce the value of older annotations at all because they're not conflicting. But sometimes changes come about because you discover what you thought was one protein, in fact, two proteins. And then you have to split all the annotations using the first protein label into two separate labels, and you don't know which way to go. So you have to do some research. But if it's joining, if you discover that two terms are in fact one entity, then it's easy, you just combine all the annotations. So then there's the splitting. And splitting occurs relatively rarely, and you can keep track of splittings, and it turns out that it's feasible. It's not always done well, but it's feasible to just go back and redo those few annotations where the splitting term needs to be redirected. So that's my answer, and I'm sticking to it. But it's not a simple problem. It's a problem that computers really do help to address.
This is a somewhat related issue, and it relates to the fact that we don't necessarily know the exact number of genes, for example, the human genome, and we don't even actually define what exactly a gene is. Can you comment on that?
Yes, so the gene ontology is not an ontology of genes, and it doesn't contain the term gene. So they don't even bother to try and define the term gene. And that doesn't seem to cause problems. So there are other is truly an ontology of genes. There's a sequence ontology, which is an ontology of gene sequences, which tells you the different types of sequences and different ways of joining sequences. So all of those work, and again, you probably would not be happy with a definition of gene that they provide, but you don't. Every ontology is going to have some terms which are, as we say, primitive. That is to say, they're not defined. And you have to have some terms which are primitive, because if you wanted to define every term in terms of other terms which are simpler, logically more well understood, then you would go on forever. There would be an infinite process, and eventually you have to stop defining and just say, everybody knows what identity means. Everybody knows what cell means. We're not going to try and define cell. Or maybe we say a cell is a unit of biological organization, but that means. Everybody knows what cell means. We're not going to try and define cell. Or maybe we say a cell is a unit of biological organization, but then we're not going to define unit. Or life. So there are going to be some terms which are just so basic that we don't try to define. So that's my answer.
So Barry, this is my one last question, and I was wondering, I thought one of the earlier statistical efforts at replication was this meta-analysis paradigm. And I think a later version, I don't know whether it was statistically motivated or whether it was motivated by economic factors, was to sort of do real-world experiments in uncontrolled settings. I don't know if you can call that an experiment, but those were the two themes that was sort of passing through. And could you sort of comment as to what you think of these two approaches, meta-analysis as well as these?
Yeah, so meta-analysis I can talk about, because meta-analysis is one of those things which becomes much easier if people use common ontologies, because then you really can compare across studies and you can find all the studies relevant to a given phenomenon if the phenomenon is described in the same way in all studies. And that's one of the reasons why the NIH is trying to get people to use registries for chemistry terms and keyword lists, why journals are trying to get authors to use keyword lists for tagging their papers. Nature Publishing Group has an ontology team, actually, which is trying to establish a definitive set of keywords for use in tagging papers published in Nature. And the more you do that, the more meta-analysis becomes, even very ambitious kinds of meta-analysis. For instance, one of my former ontologist colleagues did a meta-analysis of all NIH-funded published papers to find out which was the world's most expensive paper. And it was a paper done in Harvard. And I can't remember anymore, but it was quite an interesting experiment. Now, with regard to the other side, I don't really have anything intelligent to say. I'm a neat rather than a scruffy, so I want everything to be neat. And that's what you were describing. Sounds like something that scruffy people might like. I mean, scruffy people in the sense of neats versus scruffies. Not, you look pretty neat yourself.
If there are no further questions,